nucleotides and amino acids, nor could be drawn theoretically. There existed ample evidences to support the notion, okay, or specific notion, specific area that changes in nucleic acid were responsible for change in amino acid sequence, amino acids in proteins. Now sequentially this lead to the proposition of genetic code. Okay, how this genetic code appears that could direct the sequence of amino acids which helps in the process of protein synthesis, let us learn. DNA was very exciting. The proposition and deciphering of genetic code were most challenging in the DNA. In a very true sense, it is required involvement of scientists, okay, various physicists, organic chemists, biochemists, geneticists, all of them involved in to discover the genetic code. George Gamow, great scientist, George Gamow, physicist, who argued that there are only 4 bases if they have to code for 20 amino acids. 20 amino acids. They have to code for 20 amino acids. The code should constitute a combination of bases. Now in this case, he suggested that in order to code for all 20 amino acids, okay, the code should be made up of 3 nucleotides. What are those nucleotides? We are going to learn. This was very bold proposition that because of combination of 43 base pairs, let us imagine 4 into 4 into 4 would generate 64 codons. Okay, these generating many more codons than a required amount. Now, providing proof that codon was triplet. If you take the codon, let us take the codon. A codon is considered as a triplet, was more daunting task. The chemical method was developed by Hargobind Khorana, a great scientist, okay, in synthesizing RNA molecule. Now, with the defining combinations of bases, right, homopolymers and copopolymers, Marshall and Nirenbergs, who are they? Marshall and Nirenbergs, okay, and Nirenberg cell free system of protein synthesis finally helped to code to understand, okay, and Severo-Okhara enzyme, polynucleotide phosphorylase, was also helpful in polymerizing, okay, RNA with defined sequence in the template independent manner. Enzymatic synthesis of RNA also occurs. And finally, check code for genetic code. What is the check code for genetic code? There is a check code, okay, was prepared in the table. Okay, you can see the code here. I'd like to take 4 boxes which helps to understand the process of genetic code. Okay, 1, 2, 3 and 4. 1, 2, 3 and 4. Here I am taking uracil, cytosine, okay, adenine and guanine. Here I am taking uracil, cytosine, adenine and guanine. Okay. So here also I am taking uracil, cytosine, adenine and guanine. Uracil, uracil, and here also uracil. So 3 uracil groups code for phenyl. Okay. What is that? Phenylalanine. Phenyl. Okay. An amino acid called phenyl. And whereas Next code will be uracil, uracil and cytosine. Uracil, uracil and cytosine. Which code for phenyl? Again, uracil, uracil and adenine. Uracil, uracil and adenine. Which code for another amino acid? LEU. Okay. And whatever the amino acids you can see here, okay, all these amino acids are coded by the specific bases. Remaining part, if you see, the silent features of genetic code as shown in the figure, and the codon is triplet. If you take the codon, it is triplet, and 61 codons, if you carefully see, for amino acids 3 codons are present, do not code for any amino acid, hence they function as stop codons. Some amino acids are coded by more than one codon, hence the code is degenerate. The codon is read mRNA in contiguous fashion. There are no punctuations. The code is nearly universal for from bacteria to human. Uracil, 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 code for phenylalanine, an amino acid indicated by pH. Whereas some exceptions to this rule have been found in mitochondrial codons, mitochondrial DNA. So this mitochondrial DNA, okay, mitochondrial DNA, and in some protozoans, AUG, which is having a dual functions. A mother mitochondrial DNA, okay, a mother mitochondrial DNA and a daughter and son mitochondrial DNA will be similar. Why? Because it is occurring during the time of inheritance process. Okay, AUG code for methionine, and it is also act as a initiator codon. Whereas uracil, adenine and adenine, uracil, adenine and guanine, uracil, guanine and adenine are stop terminator codons. Okay, if the sequence nucleotides in mRNA, okay, predict the sequence of amino acid coded by AUG, uracil, 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 cytosine, uracil, uracil, cytosine, uracil, 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 etc. Okay. Now try the opposite. Following is a sequence of amino acids. Following is a sequence of amino acids coded by mRNA. Predict the nucleotide sequence in the RNA. Okay. So MET, please see the phenylalanine PHE. Do you face any difficulty in predicting the opposite? Can you now correlate with the two properties of genetic code you have learned?